All right, welcome back. Muscles, it is. I have a feeling that as far as muscles go, I'm going to attempt to break this into 3 different videos. I want to take my time with it. Actually, let me set my timer. Only because some of the terms, at least for me as a student on your side of the computer right now, the terms I found confusing: myofibrils, myofilaments, myofibers, and it took me a long time, and I still don't think I have it perfectly down as far as the order in which we find them and what the details are that allow for a muscle to contract and do some other interesting stuff. So I want to play around with some of this microscopic stuff, and then we'll take a look at actually the physiology and how do we get a muscle to contract, and probably finish up with a little bit more physiology. So that's what I'm looking for with this particular system. I think that if I was to ask you, well, what's a muscle do? You would say, well, it contracts, it moves things. It does more than that. And you might think, well, okay, so it's moving my femur, or it's moving my radius, or it's moving some other part of my skeletal system. It does more than that. It will move hairs, as you know, with erector pili. It will move food through the digestive tract. It will help to move lymphatic fluids through the lymphatic system. So movement is a big deal. It's not just acting on the skeletal system. Also, if anybody's noticed recently, your heart should be pumping. If it's not, you're probably not watching this. And if that's the case, that's contraction too. And so you're moving the circulatory system through your body. So when it comes to movement, sorry, I didn't realize how small that font was. There is a lot of material and numerous parts of our body that are moved using the skeletal system. Also, if you just stand still and you don't try and move any of the skeletal system, you realize that, oh yeah, stabilizing joints, holding a good posture is all done via muscle as well. And heat production, I threw this one on there as well. Muscles are huge producers of heat. So it's more than just moving a bone. It's a lot more intricate than that. To be able to pull all of this off, we essentially need 3 different types of muscle. And there are 3 different types of tissue. When this whole COVID thing came about, we were dealing with histology, and we were going to look at these and be able to tell the difference between them pretty easily, I would hope. So I want to address how they're different structurally and how they behave differently. So I pulled this off of the web. Here are these 3 types. Cardiac muscle, I think you can probably figure out where we find that. So muscle that deals directly with the heart. Skeletal, we're dealing with that acts directly on the skeletal system. And smooth, which we get in a number of different areas, especially through the digestive system. Now some of the big differences are these. I think the first one is smooth and cardiac are kind of what you expect. They're mononucleate. So they only have 1 nuclei per cell. You might think, well, yeah, pretty much all the cells we've looked at have been mononucleate. It gets a little weird when we get to skeletal. This is considered, multi-nucleate. So each one of these individual cells, and I don't see a nice distinctive membrane on each end, and I can count possibly 4 in there. Each one of these is going to have more than 1 nuclei, and I'll talk about how that's possible, because it's something we're going to see a lot. You're going to see a lot with the world of anatomy and phys. So structurally, few differences. Oh, one other difference. When it gets to cardiac, and it doesn't show up really well on here, we have intercalated discs that we find between the cardiac muscles, or the cells of the cardiac muscle. What the intercalated disc will do, you'll get in more of this in AMP2, is it will allow a impulse from one to move really right over to the next one without even hesitating. So intercalated discs, mononucleate, and it's involuntary. So you don't die at night because your heart stops because you quit thinking about it. It's all involuntary. So that's a look at cardiac. Skeletal. And skeletal is the one that we're going to spend most of our time on with these next few videos. Smooth muscle you'll deal with a lot when you get to the digestive system. But what we find here is, yeah, it's multinucleate. It also happens to be pretty fast. It reacts very quickly, it responds very quickly, and you'll see how in the next video or two. And it's voluntary. You decide when you raise your hand or when you sneeze into your elbow. So multinucleate, kind of weird, no intercalated discs. Oh, and it's also referred to as striated muscle. Cardiac has these weird striations too. And it has to deal with the organization of the internal structure of the cells. Smooth, no striations, hence the name smooth. Smooth muscle is very slow compared to skeletal muscle. It's involuntary compared to skeletal muscle. And as I mentioned, mononucleate. So these are the types, and like I said a few minutes ago, we're going to be focusing on primarily skeletal. So let me move up here. Here's just another image. I grabbed this off the web because this cardiac shows these nice little intercalated discs. Also, it shows you again how skeletal muscle now in the upper left is very, very multinucleate. Here's where, well, let me hold off for a minute. Let me talk about some of the things that these cells have. So all muscles will have, first of all, excitability. What excitability means is you can receive a stimulus that tells you to do something, essentially contract. If you can't receive it, if you're not excitable, you're not going to be a muscle. Along with that, along with being excitable, you have contractility. I think that's pretty straightforward. Muscles contract, which is how they'll move things. Along with that, extensibility. They can stretch. And if they're going to be contracting and they're going to be stretching, ultimately at the end of the whole process, they have to have elasticity to be able to go back to their original shape. So they can receive an input, they can contract, they can extend, and then they can bounce back to where they originally began. Now once you have all of these things, once you've got all of this that you can do, and I start to dive into the world of skeletal muscle or striated, this is what I was talking about with these terms. We look at this thing, is it a myofilament, a myofiber, a myofibril? Well, that's what we kind of have to make sure that our feet are solid on the ground when we try and figure out what is what. Take a look at the organization in here. We've got these red lines with the Z-shaped thing down the middle. Then we've got these blue lines with these little, what we call heads sticking off of them. And we see it over and over and over and over. And as we go up into 3 dimensions, and I'm going to show you an actual micrograph of this, you see these kind of commingled red and blue dots, which are these little lines that are going across the middle. You can even see them down here. So the thing that I really want to point out is the organization. When you look at this, it should strike you as, hey, that's really organized. And if I take a micrograph of this edge, this is what it looks like. And we have what are called thin filaments and thick filaments, and you can see just by the way these big blobs look, well, they're thick compared to these little things that are considered thin. So what are they? What's a thin filament? What's a thick filament? And then we have to see how they all work together to be able to put together this big motif of this organization to allow for things like muscles to be able to contract. So let's take a look at these things called myofilaments. I know this is busy. It's real busy. But I found a few images that I thought, hey, this is really good. Here's what I want to point out. If you look at this big, thick filament that goes through here, this is what we call the thick filament. And that thick filament is made up of proteins called myosin. There it is, you can read it. This myosin looks kind of like a golf club. It's got a long tail on it. It's got a couple of heads up here. And the cool thing about myosin is that that head is going to grab the thin filaments and it can do what's called a power stroke and can actually start to move past the thin. Myosin is the simple part. Actin, the thin filament, actin, the thin filament, is going to be a little bit more involved. Because ultimately what myosin wants to do is it wants to grab onto actin and it wants to slide itself past actin. It's not that easy. Let me look at this next slide. I think I'm going to come back to this one. Yeah, there's a lot going on with this. If you take a look, here are these myosin heads, and here's the little thin actin filament. So what they want to do is they want to be able to interact. But the reason I think this is more complex is we have a whole series of proteins involved, and I have to talk about what those are in my last 3 minutes or so. So, there's our actin, all these ping pong balls that are spiral. There is then tropomyosin, which is this kind of spiral corkscrew purplish color. And before I get to this whole cluster of proteins, let me explain what tropomyosin is all about. Myosin wants to bind to actin, just like I mentioned a couple minutes ago. Well, tropomyosin says, no, you're not going to bind. So what we have to do is figure out how do we pull the tropomyosin, follow me on this part of the actin, away from the binding site. In order to do that, we need this little complex called TNI, TNC, and TNT. And what I'm going to do at the very end here before I cut you off is I want to take a look at this in cross section. If I can, let me see how this drawing is going to go. So right here is our actin. Now the tropomyosin, I'm going to put here and here. So what we have on one side, we have on the other. And the tropomyosin right now is blocking the binding site. The binding sites will be right here, right here. And then we've got our TNI, our TNT, and our TNC. And they'll look kind of like this. Now, here's what attaches to what on this particular diagram. The TNT will bind to the actin. Would have been nice, well, yeah, this one is now our TNC. And TNC is going to bind to calcium. That's going to be our trigger. And then our TNT is what's going to bind to the tropomyosin. Let me make sure I just got that right. TNT to tropomyosin, TNI to the actin. Now what's going to have to happen is simply this. Once calcium binds, and you might wonder where does calcium come from, we'll get to that. Once the calcium binds here, it moves this whole complex back and it exposes the binding site for myosin. So that's what we're going to want to do. And then somehow, I'll clean this up on our next video, somehow we need to do this power stroke thing and slide these thin and thick filaments past each other, and that's going to allow us to contract. That's where we want to go before we ever get to the level of the cell, but we're getting close. So let me kill this, post it, and I'll pick it up from here with a clean board.